Sorry about that last one. That's why I have to keep an eye on the time because it will just cut me right off at 15 minutes. But anyway, we were talking about the reabsorption at the PCT. And please understand that most reabsorption, we're talking about say 70–80% of reabsorption is going to take place here. At the PCT. And what happened, this happens in various different ways, is you can have sodium can be reabsorbed with regard to cotransport or with regard to, sorry, I'm looking for a specific spot in my book. So reabsorption of organic nutrients can be based on facilitated transport. Remember, that's a type of facilitated diffusion where a protein carrier is used, or co-transport, where one thing is pumped out while another one is brought in. You can have active reabsorption of ions, particularly active reabsorption of sodium ions. But that active reabsorption can be sodium, can be potassium, bicarbonate ions, magnesium, phosphate, and sulfate ions. This would be a pump. A pumping action. Remember, or just refresh your memory, a pump is an embedded protein that requires ATP. ATP comes in and will change the shape of the protein so that the ion can fit in and then open up and be able to move the ion from one space to another. There also can be with regard to active reabsorption, that can be acted on, so that can be self-regulating, but it can also be changed or influenced by hormonal levels, and such as angiotensin II, which we just talked about. And you also absorb carbon dioxide. It basically it is done because that helps the body reabsorb bicarbonate ions. That's what you're seeing at the in the yellow area, the tubular fluid. You have hydrogen and bicarbonate ion. That temporarily becomes a compound that then can be reabsorbed as carbon dioxide and water. And the reason you want that is you want that bicarbonate ion as part of your buffer system. Water is reabsorbed in need, but there are regulations with regard to how much is reabsorbed because solute concentrations need to remain optimal inside and outside the tubes, so that the osmosis pulls the water out, but concentration levels of the solutes need to remain within what would be homeostasis in order to pull out water or not pull out water. Passive reabsorption of ions is also going to take place. You have active reabsorption of ions, certain ions, passive reabsorption, particularly if the solutes such as urea, chloride ions, lipid-soluble material will diffuse out, and other ions will actually be reabsorbed. It helps the concentration of the solutes in the fluid that's moving on down to make sure that downstream of this tube is able to function in its normal way also. So what you have here in this image, and you have this in your book, so what you are looking at is an individual item that is in the proximal convoluted tubule. You're remembering that most reabsorption takes place here. It takes place in different manners depending on what is being reabsorbed, but still the what then is the filtrate that is going to move into the loop of Henle. The concentration of that filtrate in terms of the solute water concentration needs to remain in what would be considered optimal for homeostasis as it moves through the rest of the tube system. One of the main things that we look at is anything we can use. So organic matter needs to be taken in. And then sodium is very important to be taken in back into the peritubular capillaries, as well as that bicarbonate ion. So all of them have a slightly different pathway, but this is everything that's happening in the PCT. And then moving on to the loop of Henle, that's that hanging loop, whether you are a cortical nephron or a juxtamedullary nephron, you both have a loop of Henle. And what happens is water and solutes are balanced. So there is some reabsorption of solutes, but it's balanced. For the first time, some reabsorption of water is not automatically coupled with solutes. Because what we're trying to do is to increase the concentration of solutes. As this filtrate moves from the descending loop to the ascending loop, different things will happen in each portion, and in the ascending loop, that happens because of the concentration gradient you have of the solutes in liquid solution. So the thing that does this is different permeabilities. So you have passive transport of the filtrate down the descending limb. That's the thin that is actually considered thinner than the thick ascending limb, and it is much more permeable to water. So you have independent regulation of volume and osmolarity of body fluids. Some of that is because you have these sodium, potassium, and chloride transporters. And what they do is they function in the reabsorption of sodium and chloride, which promotes a reabsorption of cations. Little or no water is reabsorbed in the ascending limb. Little or more in the ascending limb. Here we are in the loop of Henle. The thick cells of the ascending limb. We're going to look at a few slides of the loop of Henle because we're going to try to understand what this countercurrent multiplication is, the idea of it. And what's happening here, just to clarify or talk a little bit about this mechanism of sodium and chloride ion transporting, involves a carrier. So what you have is something that actually can facilitate the transport of sodium, potassium, and chloride. It's in the apical surface of the epithelial cells of the thick ascending limb, and it requires at the ascending limb, at the basal surface, there are also carriers. So at the apical, so the surface that's closest to the tube, and then the surface that's closest to the capillary bed. So you have the apical surface closest to the tube, the basilar surface closest to the capillary bed, and what you have is a co-transport. What these arrows are showing you chloride, two chlorides coming in, as well as potassium and sodium, and then what you have in the coming into the bloodstream, the chloride, a little potassium, a little sodium, but also you have potassium moving on out. So this actually helps you have the ion-cation concentrations in terms of the ascending limb, the thick portion of the ascending limb of this loop of Henle. Countercurrent multiplication, this is what I was referring to with regard to an incredible amount of water is reabsorbed in the descending limb of the loop of Henle. That causes an increase in the osmolarity. You go from 300 down to 1,200 at the bottom of the loop of Henle. That increased concentration is going to allow solutes to move out just based on a concentration gradient. It also, as it moves on up, is going to, because water is needed, solutes are going to move out so that actually is going to keep the ascending limb from being permeable to water. It's not so much that the cells are different, the concentration of solutes to water is very different between the descending limb and the ascending limb. So the descending limb over to the left, the solute water concentration is optimal for you to lose water. On the ascending limb, which is on the right, the water solute concentration level is optimum for you to lose solutes. So it actually facilitates the process. And this is the idea of countercurrent multiplication. And furthermore, what you have with regard to the capillary beds is the ability to reabsorb this. So reabsorb the water, reabsorb the ions. And then this is closer to what you have in your book. Just we're at the end of the distal convoluted tubule into the collecting duct. In the collecting duct, remember this one can be permeable or impermeable depending on the body's needs. You have it's impermeable to urea because you are trying to get urea out, but it can be permeable to water. And again, this is because of the design of the cells, but also this concentration gradient. So you have sodium and chloride would be the ones that would tend to move on out. Papillary duct is permeable to urea, so that it is going to move into the papillary duct. That papillary duct is then going to be one with the calyces, and with fluids, with water, makes up, that means that the papillary, that urea is going to be most of the solutes that come out. So as it moves down that collecting duct, certainly water is going to be reabsorbed. Water is going to make it through because we don't want to have the toxins too concentrated, but the papillary duct is actually permeable to urea, not so much to sodium and chloride, and so the urea is going to move on out, and that is part of our waste product. A little bit more about the distal convoluted tubule. There is some reabsorption in the early distal, and that's going to be sodium and chloride. This does happen to be the major site where parathyroid hormone stimulates reabsorption of calcium, depending on blood calcium levels. In the late distal convoluted tubule, you have reabsorption and secretion. And most of the filtrates have been returned now. Principal cells reabsorb the final touches on the urine, you might say, will be between sodium and potassium. So final touches is what you have here at this area. You have certain cells in that area that can reabsorb potassium and bicarbonate ion and then secrete hydrogen ions. So the distal convoluted tubule, the late distal convoluted tubule, is what would set actually the pH of the urine. So by secreting hydrogen ions, you're making the urine more acidic. The amount of reabsorption and solute reabsorption is ever changing, depends on the body need. So most action in terms of reabsorption is taking place in the proximal convoluted tubule. The composition of urine is changing based on need. from the descending limb, where again more water is reabsorbed into the peritubular capillaries, and then ascending limb, distal convoluted tubule into the collecting duct. And we'll see you on the next teaching part.